So I am the Director of Clinical Research here at the Institute, and I'm also a neurologist. I see patients in the clinic. And I'm going to give you just some sort of vignette updates on some of the work we've been doing in clinical research. So these are some of the kinds of questions that are important to us that we try to address when doing our research work. And most of these are questions without answers. So it just shows you how far we have to go. We don't know how many people or sort of what characteristics are available in the population for Parkinson's disease. We're very interested in identifying who's at risk and what the risk factors are, because this is going to be key for understanding ways to intervene and prevent or treat Parkinson's disease. We're interested in looking for clusters of people, because that might be a way to get clues to causes, especially environmental causes, but sometimes there are familial clusters that suggest a genetic form. We need to know whether more people are getting disease, whether it's constant, whether it's decreasing, because again, those are clues about what might be the cause of disease. Very important for us to know the healthcare needs, what the cost of Parkinson's, the people who are going to need services, so that we can make appropriate public health plans for that. The cause of disease obviously is critical, and prevention of disease is our ultimate goal. So these are the kinds of things that we think about when we design our research projects. And I'm going to just give you some little updates on some recent work that we're just getting some new results on. So many of you have probably heard us talk already about the California Parkinson's Disease Registry. This is a state-run initiative. So when we're working on this, we're wearing the hats of the state of California. We're not the Parkinson's Institute. Parkinson's is reportable in California, but there was no funding for it. And so we were able to secure some funding to allow us to conduct pilot projects in 2 places, one of which is Santa Clara County, where we live, and that's the part I'm going to report to you today. The other is in 3 counties in Southern California that an associate is studying. So we've been at this for a while, and we're now starting to get back some of our results that allow us to focus on some of the things we've always wanted to know about Parkinson's. So the first, I guess this doesn't know, here is in Santa Clara County. This is the preliminary data focusing on just the year in 2007. How many people could we find who had a diagnosis by a doctor of Parkinson's? And the answer is somewhat more than 2,000 people. So that means our rates here are fairly similar to what people would project across the country. The next step that we're going to do, and this is what we're just moving into now, is to take advantage of this coding that the state of California offers to researchers. That means first you figure out where does somebody live. So you give them what's called a geocode of their address. So you get latitude, longitude, and you figure out the spot in a standardized way. And then you can use that information to overlay onto other things that the state has been collecting over many, many years. This is an example of the distribution of use of a particular pesticide called Paraquat in various parts of California. And here's Sunnyvale, here's Santa Clara County, and you can see there are some areas in Santa Clara County where use is very high, and then somewhere there's almost no use at all. So we're going to look at the distribution of individuals around the county and see how that corresponds to various types of toxic exposures that the state has been tracking since the mid-70s. And that will perhaps give us some new insights, not only into maybe causes of disease, but also give us some insights into whether or not people do better or worse if they live in an area with these kinds of exposures. We're going to look not just at this, but we hope to also be able to look at things like air pollution, particulate matter, which people also think may be related to Parkinson's. So that's update 1. Following along the theme of Paraquat, in some of our earlier studies, Paraquat is a pesticide that we know is associated with an increased risk of Parkinson's disease. And we've been trying to learn more about that. So my colleague Sam Goldman and many of us have been working in a lot of different populations to try to understand the effects of different chemicals to identify causes of disease. So in this case, we know that head injury and Paraquat individually are associated with increased risk of Parkinson's. So we were able to take advantage of a population called the Agricultural Health Study, a population of pesticide applicators, farmers and their wives, to be able to look at whether or not the combination of these two things was similar, different, bigger risk, lower risk than each one individually. And so we had already looked at this population, we've examined them, we've gone out to their farms and determined they really had Parkinson's disease, and then collected really long histories of their jobs and what they did and other health information on them. And now we were able to look at the relationship between head injury and Paraquat use, and compared to people who had neither head injury nor Paraquat use, people with just one had a slight increased risk, so about 20% greater risk of Parkinson's. People who had just Paraquat use had about 80% greater risk, but if you had both, you had more than 4 times a greater risk of disease, so a really dramatically increased risk associated with pesticide and head injury, and the particular pesticide being Paraquat. So it looks like there really is something to these additive environmental effects that's important for us to be able to look at these and understand more about disease. Following along the same theme, we also looked at another aspect in the same population of Paraquat use, and here we were looking at diet, because there's been a lot of information about diet and Parkinson's. Is this aspect of the diet good? Is that aspect of the diet bad? So we wanted to look at dietary histories that we had collected from these farmers and their wives and see how diet might relate to Paraquat. So the way you do this is you ask people about what they eat, the amounts they eat, the typical portion sizes that they eat, and then you come up with a kind of a cumulative measure about dietary intake, and then you can use software that nutritionists have developed that breaks it down into things like this much of that vitamin, this much calories, that much protein, etc. And we looked at lots of different things. I'm just presenting here what really popped out in our analysis, and that was dietary intake of polyunsaturated fatty acids. And these are associated with reduced oxidative stress, at least theoretically. And all the things we've talked about so far, so Paraquat, head injury, are things that we think increase oxidative stress. So within the biology of the brain, they cause a situation that is deleterious to the nerve cells. And so in these same farmers, looking again at the relationship between people who had exposure to Paraquat and then diet, what we found was that people who had high intake of polyunsaturated fatty acids, and especially this one called alpha-linolenic acid, had lower risk associated with Paraquat. So this shows it the other way. If you had low intake of it and you had a Paraquat exposure, again, you had about a 4-fold increased risk. So a big difference in people with diets that were low in this one alpha-linoleic acid and other polyunsaturated fatty acids. So alpha-linoleic acid is something that is diets that are high in fish or nuts, vegetable oils, and maybe particularly flaxseed, which not everybody likes, but you can buy it and dump it into your food, are ones that have high concentrations of this particular supplement. So we're looking here at disease risk. We don't know if you already have PD, if it helps you or not, so that's something for future studies. But certainly this gives us an idea that there are multiple different factors that contribute to the causes of Parkinson's, and getting that additive picture all together is going to be important for us to understand the causes and maybe ways to treat or prevent. So this is changing themes a little bit. As I mentioned, one of the things we really want to do is we want to be able to prevent the onset of the motor features of disease. And as somebody mentioned in the questions earlier, we know it's a long time until people finally manifest motor features of Parkinson's disease. And we believe that there's a period where other problems are going on that you can't necessarily always observe in the body, where the process that causes Parkinson's is happening. And we're starting to think that there are actually other markers that could be symptoms of the risk of developing Parkinson's. So we think maybe Parkinson's starts not just in the central nervous system, which causes the tremor and the slowness and the stiffness, but possibly in what we call the autonomic nervous system. So the part of the nervous system that is unconscious, that controls the bowel movements and heart rate and things like that. And that it may start there. We don't know if everybody who has problems there progresses to Parkinson's or not, but we do believe that for some people at least, the first signs can be things like constipation, which then goes on to cause an increased risk of Parkinson's. But of course, not everyone with that problem does develop Parkinson's. So we're looking for multiple different features like that, impaired sense of smell, constipation, that might help us come up with what we would call a risk profile, so that we can look in a public health way at people and predict who's at greater risk for Parkinson's. And then if we have some interventions that are safe and might be effective, we could give those to these people. So this is a study, again, this time looking in Northern California Kaiser population and looking at one particular feature called seborrheic dermatitis, which is a common dermatologic problem. People get kind of flaky and oily skin, and many people with Parkinson's have it. We were curious to see if you could identify it before the onset of Parkinson's. And so we looked in Kaiser, where people have a history of all their contacts with the healthcare system, and we could look prior to the time of their Parkinson's diagnosis and see whether people had this diagnosis of seborrheic dermatitis, and then look at other people who didn't have Parkinson's but were the same age and lived in the same places, were the same gender, to see if they had it more commonly or not. And basically what we found, we compared, we had about 2,000 Parkinson's subjects, and we had about 11,000 controls, so people who were otherwise similar but didn't have PD. And they were similar age, similar gender, similar risk factors like smoking. And we found that it was a little difference, but there was in fact an increase in seborrheic dermatitis in people with Parkinson's compared to controls. And this was what we call statistically significant. So even though it was a small increased risk, because lots of people get seborrheic dermatitis, it's not specific to PD, it's one more indicator that you might be in a risk population for Parkinson's. So if, for example, a diet with alpha-linoleic acid helps to prevent, this might be a good time to start taking that particular fatty acid into your diet if you develop this kind of a feature. So that's the way we want to use this. We're just beginning this, and it's a first step study, but it gives you an idea of approaches we could take to prevention of disease. And then the last comment, many of you have heard us talk on and on about biomarkers, and one of the other really important studies that we're doing now is one that's trying to understand biomarkers for Parkinson's. So a biomarker is a marker that would be an X-ray or a blood test that would give you an idea that this person has disease, or in somebody who has disease, is the disease getting better or worse? So if you have anemia, somebody looks at your red blood cell count and they say, oh, you have anemia, you get iron supplements and the count goes up, they say, oh, you're better. We have nothing like that for Parkinson's. No blood tests, no way to measure progression, just the things that we as physicians do in the office. So that's what we're looking for. And you've probably heard us talk about this project before. It's called PPMI, or the Parkinson's Progression Markers Initiative. And this is an international study where people with Parkinson's, early, just diagnosed Parkinson's, and controls, people who don't have Parkinson's, come into our clinic regularly and allow us to assess them. We do clinical assessments, we take blood, we get particular kinds of X-ray imaging studies, DAT scans, and MRI studies. We collect spinal fluid, and we put this all into a database that anyone can access. We put it online, any scientist can go there and request it. People can make requests to be able to use the fluid. We're a little more cautious with that because it's precious, but for a valid scientific investigation, it's available to anyone. And so that study is now ongoing. We're still recruiting. So if you are aware of someone who might be interested in this, Linda Reese is the coordinator, and you can contact her or just call downstairs, and Gloria will pass you on. So I'll just end there with that update on some of our projects and take any questions.